So I'm presenting our Nature Genetics paper recently published in August. This is all come from part of my PhD thesis I did with Andrei Ryzhetsky at University of Chicago. So before we get into the details, I would like to introduce a simple model of how genotype arises, interact with environment, and kind of results to phenotypes. And this graph describes quantitative traits such as weight and height, but with a few assumptions, we can extend this to disease binary traits like diseases. So the first assumption that I would like to make is disease liability, which can be imagined as a quantitative traits that underlie the disease status. And if a patient passes through a threshold on this liability scale, the patient is affected by the disease. If the liability is lower than the threshold, then the patient deemed to be healthy. So for the first part of the study, we want to identify the impact of genetic factors and environmental factors on single disease liability, calculating heritability, common environmental effects. On the second part of the study, we want to identify their shared impact on two disease pairs and classify diseases using the genetic environmental correlations. So now I would like to give some introduction to the data that we used. So we have about 150 million patients diagnostic records between 2003 and 2011 from a Truven Market Scan database. So this is about a third of the US population. And in order to kind of differentiate and separate the genetic from the environmental effects, we have to leverage the family information stored in our data. So we start with 40 million families in our database. Of them, less than half have children's. And in order to make sure the phenotype we observe in the parents' generation and in the children's generation are comparable, we kind of limit our study to families with children age 16 or older. So basically, family with young adults. And furthermore, we want to make sure that our observation is consistent across time. And also because some of the diseases such as cancer take longer time to observe. So we want to make sure that we have the most complete diagnostic records of these patients. So we set up the enrollment at minimum of 6 years. On average, the family is in the database for 6.5 years. So similarly, we filtered on the disease of choice. So first we rule out the Mendelian diseases and make sure the disease prevalence is higher than 0.2% in both children and their parents. Then for the pairwise diseases, we further filtered on whether we can have sufficient non-trivial pairwise analyses. And also, because kind of the computational burden on this study, we kind of limit our study on 4 biological systems for the pairwise analysis: the neuropsychiatric, the cancers, immune system diseases, and also cardiovascular diseases. So next, we sort of build our model assuming the variance of disease liability is the sum of additive genetics, a couple common environments for the parents, and common sibling environments, common environment for the whole family, and also unique environment. And the heritability is calculated as the corresponding environmental or genetic effects over the total variance. So here is the result of our heritability analysis of 149 diseases across 20 biological systems. So we order the disease by their heritability estimates. The diseases are colored by their biological systems. Acronyms is in the paper, but some of them you can read easily. And for all the diseases, whatever is new that was not reported before, we mark it with a star. And there are also an error bar, even though we have very tight estimates of the errors. So if we zoom into the higher extreme, we can see that autism is the highest with the 92.4% heritable, And a couple more neuropsychiatric diseases are highly heritable as well. Several immune system diseases are highly heritable, as well as metabolic diseases. So that seems to be a trend. And if we go to the other extreme, lipoma, which is form of a benign tumor, is about 3.8% heritable. And this is the only one that have a heritability that's not statistically significant. Basically, the confidence interval past 0. Also, you can see that on the lower end of the heritable diseases, a lot of them have very low heritability. So, so this is the single disease analysis, and then we also kind of search the literature and then find 65 disease heritability estimates that was reported before. We sort of plotted against our estimates. In general, the agreements are pretty high. About 52 out of the 65 agree with previous literature. Overall, our estimate tends to be slightly lower. My guess is that since we accounted for several common environmental effects, so this is the what we find for disease heritability. Now, so we have about 84 new estimates, and the majority of the estimate that was reported before seems to agree, and the slightly lower heritability estimates. And so we go back to this liability model for single disease, and if we extend this to 2 diseases, we can see that the red dots here represents patients that have disease A. The blue dots are patients that have disease B, and the purple dots patients that have both diseases. The green means the patients are healthy. So this is showing the disease liability correlation about 0.7, and this is on the phenotypic liability level. And if we see 2 diseases that are negatively correlated, we'll see there are fewer patients with both diseases. So this is a pattern that we're sort of estimating. And similarly, with the same model, we now can estimate genetic correlation and environmental correlations. So here is the sort of the correlation matrix. The top upper right part is the genetic correlations, and the bottom left part is the environmental correlations. So red representing positive correlation, and blue representing negative correlations. The white cells you see corresponds to a correlation estimate that sort of overlapped in their confidence interval with 0. And the size of the square indicates the statistical significance of the correlation. The stars measures the 1% false discovery rate. If it passes, we put a star on the square. So you can see the majority of the correlation in our analysis is significant, different from 0 at 1% false discovery rate. And on average, from general pattern of this matrix, you can see that genetic correlation between diseases tend to be higher than their corresponding environmental correlations. However, genetic correlation and environmental correlation are themselves positively related. About the coefficient is about 0.5. And if we zoom in onto the cluster of neuropsychiatric diseases, we can see that they are very closely linked, related to each other, both genetically and environmentally. And kind of contrary to the general pattern, for neuropsychiatric diseases, both genetic and environmental correlation are about the same magnitude. This is sort of the very visible pattern we can see on this matrix. So now I will switch attention to some kind of disease pairs that have the opposite genetic versus environmental correlations. So for example, for ADHD versus two kind of skin cancers, the genetic correlation is positive and the environmental correlations are negative. And the reverse of that, it's with schizophrenia, substance abuse, type 1 diabetes, hypertension versus the two skin cancers. In this case, the genetic correlation is negative, while the environmental correlation tend to be positive or non-significant. So we also collected literature on the GWAS genetic correlations from the previous estimates of the disease pairs that we have. And we find there is a very high degree of agreement between the two estimates. So you're looking at the degree, the proportionality constant of the line is about 1.08. So it's just a straight from zero. So 45-degree line. So this indicates that the family-based genetic correlation we estimated reflect predominantly common variants. So the common variants genetic correlation and the family correlation estimates about the same trend. So with these correlations, one natural thing we can do is basically we can cluster them based on different correlation patterns. So here is the classification we derived from the genetic correlation versus environmental correlation. First, you can see that we sort of highlighted 6 very stable clusters, meaning these diseases stay together regardless of the factors we kind of interested in. So the first cluster is bipolar and mood disorder, both in genetic and environment. Then, somewhat surprisingly, the type 1 diabetes and general hypertension is also closely clustered in both factors. The other ones, the chronic sinusitis is a sort of inflammation of the nose sinus tract, which is very close to related to the upper respiratory inflammation. And the two inflammatory disease about the guts, closely related, skin cancer, close related. So, so we generated these classifications, but we don't know how it sort of compared to what's usually used by the doctors. So we also, in a way, generated the ICD9 disease classification tree. So here you can see all the immune system diseases, inflammatory diseases cluster tightly together, cardiovascular diseases cluster together, and eye inflammation is, and migraines are the disease of central nerve sensory organs, and all the neuropsychiatric diseases are in the mental disorders categories. So this is what ICD9 classifies diseases. So we sort of compare our analysis classification with what's already available, and we see that there are a couple kind of stable clusters that are surprising. So within the neuropsychiatric disease, we see anxiety and depression have very stable and highly correlated genetic correlations. And that is kind of contrary to how anxiety and depression is classified in the ICD-9 code. As we mentioned before, type 1 diabetes is usually known as a immune system disease, is tightly clustered with hypertension. Also, a migraine is tightly clustered with IBS. Maybe I should mention a little bit about a number on the cluster. So each number represents the percentage of times that we regenerate the classification, and this cluster show up over and over. So for migraine IBS, we tried 10,000 times iterations, and about 89% of time the migraine and IBS cluster together. in the genetic classification. Similarly, in the environmental cluster, we have some surprising findings, and mostly the surprising one are type 1 diabetes with general hypertension, and the migraine with IBS, and also migraine with another kinds of inflammatory diseases. So these are very surprising coming from genetic and environmental analysis. So to recap, we estimated genetic correlations across all pairs of the 29 diseases. We have a linear relationship with a proportionality of 1.15 between the total liability and genetic correlation. So this is kind of reported before that in some ways people can infer genetic correlation from the total liability correlations. And we sort of confirm that with the literature. And the genetic correlation obtained by two different methods, GWAS and our family analysis, kind of agree well. So yeah, I would like to kind of going quickly, but acknowledge the support from my advisor, Andrew Rizetsky. Hallie also helped on this project, Nancy Cox from Vanderbilt, and also Hui Feng from Microsoft. I'll be happy to take any questions